Hi everyone, my name is Brianna Jackson, and this is my synopsis of the podcast Prokaryotes Promote Past and Parent Peculiarities. The host of this podcast is Jesse Nowar, and the contributing doctor that spoke in the podcast is Dr. Per Steinberg. The paper that was analyzed in this podcast is The Gut Microbe Participates in Transgenerational Inheritance of Low Temperature Responses in Drosophila melanogaster. And Drosophila melanogaster is actually a type of fruit fly. And this is the type of fruit fly that will be experimented on later on in the discussion. So, to begin the discussion of the microbe itself, the microbe of the episode is microbe, meaning a microorganism that can cause disease or it can cause fermentation, is Bifidobacterium breve. And if you break this down by the root word, bifido means split, breve means short, therefore Bifidobacterium breve is a short split rod. The microbe was discovered in 1963 and it was found in the poop of human infants. And Bifidobacterium breve is actually something that is needed for us to break down milk sugars that we can't break down on our own, called galactico-oligosaccharides. And this microbe actually comes from human breast milk, but it is not for the purpose of feeding the baby, but it is for feeding the baby's intestinal bacteria. So the microbe does come from human breast milk, and it is passed from mother to child, and it is considered to be a probiotic. This microbe can also be used to treat some intestinal diseases such as IBS and different things like that. So now we're going to discuss the paper, and Jesse begins the discussion on explaining how adaptations are related to natural selection. So natural selection is the process whereby organisms better adapted to the environment tend to survive and produce offsprings more than the ones who aren't. The part of the species that is better adapted to the environment are going to be the ones that are more successful in reproduction. Therefore, their already better equipped genes are going to be the ones that are going to be passed down as well. So, when there's no technology at all involved, usually most of the adaptations in species come from just the genetic makeups. And not all inheritance are products of a genosequence, but they can be characteristics of that individual as well, passed down from parent to offspring. So Dr. Steinberg weighs in on the discussion by saying that recent studies have shown that, for example, an altered diet can be something that is passed down. So this could be in certain countries, certain cultures eat certain things. So this would be an example of an altered diet that has been passed down from generation to generation. A quote from Dr. Steinberg during the podcast related to this matter is, "Responses to an altered environment does not get encoded into the DNA, and the mechanisms for this inheritance has actually been hard to find." So he's mentioning the inheritance for an altered environment. The reason he's mentioning an altered environment is because later on in the discussion, they did perform an experiment on fruit flies, and they tested whether or not they could pass down cold adaptations. So the mechanisms that Dr. Steinberg is referring to is epigenetics. Epigenetics is the study of changes in organisms caused by the modification of gene expression rather than the genetic code itself. So what this basically means is that the same gene can be expressed in two completely different ways. So if, for example, the same gene were in two different individuals, it can have two different ways of being expressed in that individual. It's not really a constant thing. An example of inheritable traits would be considered cultures, not the diet, but cultures and technology. And this can be passed down from family and across a family as well. So Dr. Steinberg and his colleagues found that fruit flies can pass on adaptations to cold temperatures actually through their gut microbes. Gut microbes are a population of microbes that's living in your intestines, and they did publish their findings, and that's what one of the things we'll discuss later. The reason for this study is the field of microbiome transplantation or fecal transfer has been getting a lot of attention from the medical field lately. These processes actually look to be a promising treatment for a number of intestinal diseases, as I mentioned earlier in my introduction. Dr. Steinberg then continues to tell how he was reading a study one day, and it was on the use of fecal transfer to actually treat a specific intestinal disease. So the woman who received the fecal transfer was successfully treated for her disease, but she then later became obese. After research, it was discovered that the donor of the fecal matter that was transferred was actually obese as well. This incident helps to prove that unrelated traits can actually be passed down or can sometimes be transferred with the microbiome, and it can be inherited from parent to offspring. It wouldn't be considered a far-fetched idea that the microbiomes could play a role in the transgeneral inheritance of environmental changes as well. So, after coming to this conclusion, Dr. Steinberg and his colleagues decided to conduct an experiment using fruit flies. So, the reason that they chose fruit flies is because they lay their eggs in the same place that the food is available to the baby flies for them to eat. When the larvae hatch, they feed on the fecal matter of the parent flies, and the matter that they consume is full of the parent's gut microbes. Therefore, the source of where the offspring is getting their microbes could actually be easier to control because they feed on the fecal matter of the parents. So, on to the discussion of the experiment. So, they started with growing two different sets of fruit flies. Well, they were both the same species, but at two different temperatures. They grew one at what is considered to be their natural temperature of 25 degrees, and then they grew one at 18 degrees, which would actually be considered a cold shock for them. So they took eggs from both of these flies and cleaned them and put them in a clean environment and everything, and they placed clean food and added feces from the flies that were grew at either temperature, whether it be the 25 degrees or the 18 degree temperature. They put fecal matter from both of those flies into the place where the larvae was going to feed. But none of the actual larvae had experienced an 18 degree weather because they were all grew at their natural temperature of 25 degrees. To verify the transfer of flies, they sequenced the microbes with a 16S sequence. The 16S sequence relies on sequencing of the 16S ribosomal RNA gene as the genetic marker to study bacteria. And the genetic marker actually has hypervariable regions used for bacteria identifications. And this process also helps to identify the flies that were adapted to the different temperatures. These two different microbes at the different temperatures were accurately able to be transferred to the baby flies for consumption. Jesse then begins to explain how there are two different families of fecal bacteria that were passed on. from these flies. One being Acetobacteria ACA, which would be considered similar to acetic acid, and one being Lactobacillus ACA, which is similar to lactic acids. Both of these two different bacteria have different metabolisms. They found that in the natural temperature of the flies, they had equal amounts of both of these bacteria, and in the cold temperatures, they really only had a large amount of the Acetobacteria ACA. Although they did have this finding, they're not really sure what this exactly means for their data. But they did find, looking at all levels of the genes that were expressed, that there was a clear inheritance of adaptations to cold temperatures. But the greatest effect was to the eggs, which shows a clear contribution of the microbiome, which helps to show gene expression from parent to offspring. And the examiners did a pretty good job of weighing out all the different factors that could have affected these gene expressions, and they came to the conclusion that really only the microbe could have affected these genes like this. So this actually opens up a door for a future study that is, can microbes be passed down just from generation to generation, or does it really just stop at the one that they tested? Next, during the experiment, they looked at how specific genes can be affected by the microbiomes in cold temperatures. So they made a chart that showed the effective genes more in cold temperatures and the warm temperatures, so you could see everything. So the genes that were upregulated or expressed more were done pretty evenly throughout the fly, especially when focusing on the middle of the fly and just the makeup of the fly itself. But the ones that were actually downregulated or less expressed were done so more in the gut or the intestinal region of the fly. This would actually be assumed, considering this was an experiment to test the gut microbiomes of the fly. So this would actually be something that they would look for. They actually did not prove that the microbiomes were the only factor that affected these flies. Factors that would be considered against the idea of it only being the microbiomes that affected these changes would be the overall treatment of the flies themselves, and could it be that the eggs that were from the 18-degree temperature were already adapted? Those are two things that could have affected these results as well. But they actually did a pretty good job of trying to rule out every factor that could have been involved in affecting these microbiomes like that. So overall, we do not know how this will affect the overall life of the fly itself. We don't know if it's going to carry on a normal cycle of life, if it's going to have any intestinal issues, or if anything like that is going to happen to the fly. But we do know that adaptations can be passed down through their fecal matter from parent to offspring. So, as a summary, the fruit flies did transfer adaptations to cold weather and can express the gene expressions of adaptations. So, I guess that they could have said that their experiment was a success, because they did find that adaptations to the 18-degree temperature was passed down, and different bacteria in the intestines were of different levels with different flies. But this also leaves open a discussion for future studies with more than just flies, maybe humans in addition, such as drug addiction, obesity, mental disorders, or adaptations to humans as well, because all over the world there's different temperatures, so maybe humans can get adapted to that as well. So thank you to everyone for watching my video, and I hope you enjoyed it. I hope I did a pretty good job of explaining the podcast to you. And thanks.